Hello there, my name is Dawei Liu, and I'll be presenting on my proposal titled Ratiometric Combinations of CAR Immune Cells for Solid Tumors. Solid tumors, such as sarcomas and gliomas, are some of the most detrimental and common forms of cancer with high mortality. The central obstacle that my project seeks to overcome is the poor effectiveness of cell-based chimeric antigen receptor, or CAR immunotherapy, due to the immunosuppressive tumor microenvironment. CAR immune cells are cells harvested from cancer patients or donors that are engineered to express a certain receptor that binds to the cancerous cells and kills them. However, while they've demonstrated remarkable effectiveness in liquid cancers, they've struggled to perform against solid tumors. They can induce a number of life-threatening side effects, largely resulting from the immune system's reaction to the CAR immune cell infusions and the inability of the CAR cells to distinguish between receptors on healthy and tumor cells. The tumors also employ numerous advantages to prevent CAR immune cells from killing, such as by releasing chemical signals that tell the immune cells to become inactive. They're also naturally more difficult to target due to the lack of suitable antigens expressed on their surfaces and being difficult to infiltrate. Rather than administering these products individually, my proposed solution revolves around combining each effector cell, or also known as simultaneous administration or co-administration. It is assumed that each cell type would have their own profile with distinct advantages and disadvantages in their mechanism of action, so by administering them together, you would compensate for their weaknesses. This diverse composition of cells, or heterogeneity, has already been demonstrated to be effective in lessening dose-dependent adverse side effects by reducing the number of cells necessary to achieve the same effect. The use of multi-targeting CAR cells along with other synthetic products can supplement monotherapy, or a 100% ratio. Further refinements, such as incorporating the use of computational algorithms and single-cell spatial analyses to model how the tumor would look in 3D, could be used to predict and optimize the appropriate dosages of CAR immune cell combinations to administer. The potential combinations would be endless due to the nature of how they could be combined, such as dual targeting with single targeting, the number of splits, the potential target antigens such as glycan or sugar targeting and CARs, and the number and timing of infusions that could reshape the environment surrounding the cancer cells. I believe that this would work due to the immune system naturally being designed to work together, and harnessing this synergy could be validated using cytotoxicity and other assays. The main limits of this cocktail approach would be the potential toxicities resulting from crosstalk or unforeseen interference of one cell type with another. Tumor antigen escape or loss could also lead to relapse, and the added complexity of this microenvironment would be difficult to model and thus difficult for regulatory bodies to approve. It would also be more complex to manage clinically and labor-intensive and expensive to manufacture, but I believe that these prohibitive factors and shortcomings may be addressed using novel alternative expansion methods. Here are my references. Thanks for watching.